So, hi everyone. We're last group, so bear with us. So, I'm Evan.
I'm JD.
I'm Ryan.
Oh, sorry.
It is possible to get two votes.
Yeah, so today we'll be talking about how to make nanoparticles, specifically gold nanoparticles. So, as you all know by now, nanoparticles are used in like a lot of stuff from cancer treatment, electronics, and like stained glass, and a whole bunch of other stuff. So in our experiment, we made gold nanoparticles by like getting gold in solution, which like they're in ions. Then you add like a reducing agent. So you add electrons to them to neutralize the atoms. So then after that, you heat up the solution so that the gold atoms will stick together to form like a nanoparticle. And once it changes color, then you like add the capping agent, which is citrate, to like stop them from getting too large. So on to the analogy.
Okay, so I'm going to start analogy from the bottom right. It's like they are saying, remember old jewelry jacket, and what's that? Other sharp object must be removed to enter the play area. So it's like saying you need to be reduced first because they have the same positive charge. So like they place a magnet, both north side together, they'll repulse each other. So we need to take out the charge. They are neutral, so they can come together to form a nanoparticle, and that is the top right corner doing. So they are reducing them, take out the positive charge jacket, and then the middle part is the forming of the nanoparticle. So the hamster ball is the capping agent. They prevent them to come together, and if they don't have the hamster ball, they will come together like these two guys. And the bottom left is the next step we are going to do is the forming the nano star. And on the very top right corner, the sponsored by California Orange means that both the citrate act as both reducing agent and the capping agent. Yeah.
Yeah. This analogy really describes really well and in depth the process of making what we called the seeds, which were the baseline gold nanoparticles that we made. But also, in addition to doing that on the same day of this lab, we also, what we did is we made 3 different kinds of nanoparticles of different size and shape to examine the effects of light on these nanoparticles. So besides these gold nanoparticles, as you can see by the +3 charge, we also made nanostars, and we also made slightly larger gold nanoparticles, and each of these interacted differently with light. And this was all in the scope of one lab. And that has to do a lot with plasmonics, which a lot of groups already went over. So essentially, light is an electromagnetic wave. These nanoparticles were all made of metal, which have like a sea of unbound electrons that can oscillate if exposed to an electromagnetic wave. Essentially, different nanostructures and nanoparticles are on different size scales, and so is light. And so the different sized and shaped nanoparticles the electron field will oscillate in sync with the light waves, so that particle will absorb that type of light as opposed to all the other types of light. So the other type, you see more of the complementary type of light when that color is absorbed, so it gives off, you see a different color. So that's basically what happened with the gold nanoparticles. We saw red because green light was absorbed. But then later we also made larger gold nanoparticles so that those nanoparticles were, this was like kind of freaky actually. It was these nanoparticles were big enough that were big enough that they had a higher percent chance of scattering light as opposed to just having light transform through it. But they were still small enough that they only absorbed green light. So we were able to see if you shined a light directly at the solution, it scattered light back at you and you saw green because red was absorbed, or it was actually brown because there was red still. And then if you look on the other side where the light was, then you saw red because it was transformed through the solution. And so with the nanostars, we saw blue because there were the tiny arms of the stars were a lot smaller, so they absorbed another kind of light that, and in some cases, with the arms were shorter, so that meant that there was more of just the sphere base size. It was kind of a purple color that was in between blue and red. Yeah. So nanoparticles are pretty neat, and they interact with light in cool ways, and this analogy shows how you make them. Thank you very much.
Okay, we got to end our questions on a strong note.
Pressure. Okay. So a lot of people so far have mentioned citrate as a reducing and capping agent, which it is. But was citrate always used as a capping agent in every experiment you did?
No, citrate was not always used as a capping agent, mostly because citrate is not the best capping agent. So what we did in another part of this lab actually was to use hand soap as a capping agent instead. And then so we used hand soap to create another layer of capping agent around them. And then we took that solution and the citrate capped solution and we exposed both of them to salt, and just the presence of the ions that were dissolved from salt actually completely they ruined the citrate coated solution. So those gold atoms aggregated, and it wasn't red anymore because they got too big for the light. But on the soap coated nanoparticles, the salt didn't have any effect, and we still kept the red color.
Hey, parents, this is your last chance. Your last opportunity.
Here's the billion-dollar question. You mentioned about its potential application for cancer drugs. Okay, how can you make a nanomolecule potentially to be a cancer drug that target cancer cells in the body? I'm not looking for an answer because it's $1 billion, but what do you think? What do you think? All the experiments, everything's excellent, all the things you've learned in 1 week, but how can you use that to apply to real life and medicine?
So another group kind of talked about it. It's called the Photothermal Effect. We'll basically use gold nanoparticles. You cap them with antibodies that are specific to cancer cells, and you I guess do you inject them? I don't know. We just insert into the body, and then you shine infrared light. So it's basically red light on your arm or wherever it is. So basically the light will pass through and excite the gold nanoparticles, and the heat will kill the cancer cells.
Yeah, and I'm pretty sure they actually are already doing some kinds of experiments with this practically in rats. So they'll embed a chip underneath the skin of the rat, in the bloodstream or something, and then they'll shine a laser on it, or that may have been for something else. But yeah, essentially you have either a chip that you go in and then you shine a light from the outside, or another option was taking almost, I think it was a loop of blood, and then there was a little filter that it went through with the gold nanostars on it, and then those would kill the cells if it's cancer in the bloodstream or something like that.
There's lots of different clinical trials. The problem so far is that If you don't kill everything, but you do let them loose in the blood, then it can be worse. And so that is the challenge. It kills a lot of them, but if you don't kill all of them, it's not a good treatment yet. So I think that's why it's not a real therapeutic yet, but the method as a general idea works. You just have to make it a therapeutic.
There is also another form of using nanomaterials to treat cancer that is not based on the photothermal effect at all, but is similarly, you're using nano-sized materials for treatment. So do you remember what that might be?
Was that the silver again, or biotoxicity? No, okay, yeah, I thought. We were talking also about like lipids and how they can take different forms. So you can either have like a monolayer, you might have a sphere of them. What could you do if you had a sphere that you make that's nano-sized? What could you do with that sphere?
Oh, do you like inject like chemotherapeutic drugs in them?
Yeah.
Yeah, so if the spheres are hollow, what could you also put in the mix so that what could form inside of those spheres or be inside of the spheres?
Cancer-killing drugs that would so you'd make these spheres that would specifically target areas in the body, so you don't have to submit your entire body to like really destructive medicine.
So I just one more question about the picture. Okay, so the hamster balls. You guys said something about this, but I think this is actually really a cool analogy to the real chemistry. So can you just say a little bit more about what the hamster balls are doing?
The people are, it's kind of like an amusement park. The people are running around in these hamster balls, but the hamster balls are keeping them from sticking together, whereas like otherwise they have to hold hands to make sure they're safe. So basically they stick together. They're more attracted to each other, whereas they can't stick together.
So chemistry-wise, what might have been, the people are we're talking gold, not silver, the gold ions that are getting ready to grow on to the growing nanoparticle. In your experiment, what made the hamster balls?
The citrate made a hamster ball, but the gold ion, they don't have the citrate balls. They have a very high surface area. So the surface energy is also high, so make it very reactive. They tend to come together.
Yeah, but that's exactly right. The capping group that caps the surface can also sort of grab single ions and change how fast the whole particle grows. That's why making nanoparticles is so hard, because everything does the same things. Okay, do we have any last questions? If not, let us thank this group and can